Okay, welcome to this video. And on this video, I wanted to discuss about the physiology of the respiratory system. So we'll talk a little bit about the different phases of respiration. We'll talk about the process of breathing in and breathing out. And we'll also have a look of lung volumes, which allow us to calculate lung capacities. And we'll finish off by talking also a little bit about the lung disorders. So let's make a start by looking at our phases of respiration. And there's really four elements or four stages that take place. So the first phase is known as pulmonary ventilation. Pulmonary ventilation refers to moving air in and out of the lungs. So this is what we would commonly call the process of breathing. So that's bringing the air from the external world all the way to the alveoli. So those little air sacs at the end of the respiratory tree that we just saw in our previous lecture video. We have a stage known as external respiration. This is where gas exchange takes place between the pulmonary blood and the alveoli. So oxygen is loaded into the blood and carbon dioxide is unloaded from the blood. So that's the first time that we see gases moving between the cardiovascular system and the other parts of the body. So external gas exchange. We're also going to see respiratory gas transport. This is the transport of oxygen and carbon dioxide via bloodstream. So all of the things that we talked about when we looked at the cardiovascular system. And the final phase or event that we see in this series would be the internal gas exchange. So this is the second time when the gases are exchanging. And here the gas exchange is taking between the blood and the tissues or cells in the systemic capillaries. So now we're offloading oxygen from the blood to the tissues and picking up CO2 from the tissues to the blood to bring it back again to the lungs. And obviously these steps can go in this order, but we can go in reverse as well. So those are our four phases of respiration. I also wanted to talk about the concept of pulmonary ventilation, which is really the mechanical process that depends on the volume changes in the thoracic cavity. And the volume changes then, in turn, lead to the pressure changes, which lead to the flow of gases to equalize the pressure. So we're going to look at this concept, and I do have slides there, and I'll use those to illustrate a lot of that. But in addition to that, what I have here is a syringe, just a typical syringe that I've taken from our clinical practice, but I've cut off the very top, and I put a balloon there. And on this illustrations, I'm going to use the syringe, the inner space within there represents our thoracic cavity. And we'll see what happens as we move our thoracic cavity volume, as we increase it and decrease it, what happens to the movements of the air. And the movements of the air we can study then with the example of this balloon that I've attached there to the very top. So let's have a look what we can learn about the process of breathing. And there's really two phases to the pulmonary ventilation. So inspiration, or sometimes known as the inhalation, is the process of breathing in, so flow of air into the lungs. And then we're going to have a process of expiration, so exhalation. So this is the process of breathing out. And here, air leaves the lungs. And we're going to explore each of these in more detail, also paying attention to the structures that assist us to do this. So breathing in is an active process. So what we do when we breathe in, we make our chest cavity large. And how we do that is really due to two sets of muscles. We have our external intercostal muscles. Remember, they're between the ribs. And as they tense, they kind of lift and pull forward into the sides, the chest cavity, making the chest cavity bigger. But even more importantly, the main respiratory muscle is going to be our diaphragm muscle. Diaphragm muscle is usually kind of dome-shaped muscle separating the thoracic and abdominal cavity. And as we breathe in, this dome-shaped dome-shaped muscle now flattens out. And as it flattens out, it makes our thoracic cavity bigger. So let's have a look what happens when I make this simulated thoracic cavity bigger. So this leveler that I'm pulling would be a good analog, for example, to the diaphragm muscle. As I make it bigger, air is flowing in, as you can see by this ball tilting in. So we make our chest cavity bigger, air flows in. Let's have a look at the process of breathing out. This is a passive process. Here, the chest cavity shrinks to its original or even smaller size, and air flows out. And this is thanks to two things, elastic recoil and surface tension. So what we could see is as we relax these muscles, chest returns to normal stage. Same goes for this diaphragm muscle that's now flattened. We relax it, it forms this dome-shaped shape. And as I do so, air is flowing out of the lung, or in this simulated lung of the syringe. We can also do something that's known as forced expiration. So pushing air out beyond our normal outburst breathing. So remember, breathing in was active process. Breathing out was passive as I relax everything. And then I can go, here I recruit our oblique abdominal muscles and our transverse abdominal muscles, and also our internal intercostal muscles to really push that chest cavity as small as possible. And as I push it as small as possible, air is going to be flowing out. So I hope that that helped to illustrate the process of pulmonary ventilation a little bit. Of course, in addition, we have a bunch of other times when we move air that are not related to the breathing per se. I'm happy to discuss more about those. If you have questions, let me know and we'll unbundle some of those together. And now I wanted to next talk about the concept of lung volumes and lung capacities. So let's have a look at that. Respiratory volumes are what we measure, whereas these can be then used to calculate respiratory capacities, which are really the clinically meaningful measures for us as we do clinical work. And to do this work, we use a method and apparatus known as spirometer or do spirometry. These are very simple spirometers that are just basically plastic tubes with a leveler that moves up and down as you breathe into it, or it could be highly computerized technical apparatus that actually draws this kind of a trace over time where you see air moving in and out over that. And that's actually what I'm showing here on the next slide of spirometry results. And we're going to unbundle different sections of this chart of breathing of an individual. So let's start with a section known as tidal volume. Tidal volume, or often abbreviated as TV, refers to the normal breathing. And that's the smallest wiggly line that you see there right at the beginning and right at the end. Only about half a liter, so 500 millimeters of air is moved in and out during our normal breathing. So you see that it's a very small amount of our total lung capacity that we really tap in our normal resting breathing. So that breathing, that movement of air was known as our tidal volume. Then we have our inspiratory reserve volume known as IRV, and this is how much air you can breathe in beyond the normal breathing. So you do your normal breathing and then you take a big breath of air in. And this is, in this diagram, this upward peak that you see. It's about 2,100 to 3,200 milliliters, so 2 to 3 liters roughly, that we can breathe in. So a lot more capacity than what we use in our normal breathing. So we can breathe in a lot more air than we normally would breathe in by recruiting this inspiratory reserve volume from our lungs. Then we're also going to see our expiratory reserve volume, so often abbreviated as ERV. This is how much air in you can breathe, or how much air out you can breathe beyond your normal breathing. So I can also go have my normal breath and then go, And in this diagram, this is this downward peak that we see. So about 1200 milliliters, so a little more than 1 liter. So this was the forced breathing out. So how much air we can expel beyond the normal breathing. We have capacity there as well, beyond the normal one. And we're always going to have some residual volume, abbreviated as RV. So some air will always remain in the lungs and the respiratory pathway to keep the respiratory pathways and keep these alveoli open. If we emptied all the air, it would be like vacuum sealing a food product. And once alveoli collapse, it's really hard to undo that work or undo those collapsed alveoli. I really included this slide simply as a summary of what we've said, since I have less ability to use pointer on these recordings. You see each of these are tidal volume, inspiratory reserve volume, expiratory reserve volume, and a residual volume labeled there on the right-hand side of that trace. And now we're going to take these lung volumes that we could measure, and we're going to calculate lung capacities based on those. And remember, it is these lung capacities that we really use in clinical practice to assess patients' ventilation and how their lungs work. So let's start by looking at the inspiratory capacity. Inspiratory capacity, so IC, is calculated by adding together our tidal volume and our inspiratory reserve volume. The next measure that I've highlighted there is going to be our functional residual capacity, so FRC. This is accomplished by adding together our residual volume and our expiratory reserve volume. The next measure that I'm highlighting there is going to be our vital capacity, so our VC. This is calculated by adding together tidal volume, inspiratory reserve volume, and expiratory reserve volume. And this is the one that you want to know for the lab. So this is basically how much you can breathe in and out, both during your normal breathing and also forced breathing in and out. So really tells us the total movement of the air. We're also going to be able to calculate total lung capacity. This is calculated by adding together all of these measures, so tidal volume, inspiratory reserve volume, expiratory reserve volume, and our residual volume. So lots of things that we can do in terms of measuring ventilation, and that's going to be what we're going to be looking at in our lab later on today. I'm going to finish off this recording by briefly right here, and we will then have a very brief recording where we'll quickly talk about lung disorders in the subsequent recording. I just don't want to make this too long at this time. So let's wrap it up here for this one, and I'll see you at the next recording for lung disorders. Until then, thank you and bye.